What do you think, Alex? Maybe, I mean, we have quite a lot
Yeah,
of people already. I guess they can slowly come in.
Mm-hmm.
I'd like to just ask everybody to turn off their video so that it's not distracting for people. And also it will let the presentation run smoothly. Okay, great. Okay, so I guess I will start by introducing our speaker today, Dr. Alex Hoffmann. And thanks for speaking, you know, in our brand new seminar series on COVID. And really, my intro will go on forever about Alex, but I will try to keep it short and sweet. We all know that Alex is very talented and well-rounded with exceptional training record. And what really impresses me about Alex is how he's been extremely successful in running a huge research group with both computational and experimental components for many years. He actually got his a bachelor degree at Cambridge in physics at first, but then he got interested in biology. So he got converted to the dark side and did his PhD work with Bob Roeder at Rockefeller University. Then he did his postdoc training with David Baltimore, where he first focused on HIV. And there goes, you know, the virology interest from the beginning. And then he aimed to understand a dynamic control NF-kappa-B signaling network, which is still a major theme in Alex's lab. Alex's lab aims to study how different pathogens and stimuli elicit different signaling dynamics and how these can be changed during disease states. And I think he will go more into that today in his talk. He first set up his lab at UCSD and then was recruited to UCLA in 2014. Lucky for us. At UCLA, he is the Asher Professor of Microbiology in Microbiology, Immunology, and Molecular Genetics Department. And he's also the director of QC Bio, which has been extremely beneficial for the research community to have here, especially when a lot of our work are moving towards systems approach and bioinformatic analysis. And I'm sure that Alex's lab is very hot recently because now we're all doing remote research. He is also a great colleague, collaborator, and mentor, especially to us young faculty. So please take it away, Alex.
Well, thank you very much, Melody, for this very kind introduction. And I actually congratulate you and Brigitte for putting this seminar series together. I'm so enthusiastic about it that I volunteered and got into this first slot. So I'll talk a little bit about innate immunity, which is of interest to my group. And the talk is, you know, is not a typical research talk of the latest studies that we do, but it does, I'm trying to relate it a little bit to COVID as far as I know. And I'm trying to give some overview of the key concepts of innate immune memory and innate immune trainability, while also highlighting some of the things that are going on in my lab, because of course this is an intramural seminar series, and I'm hoping by presenting that maybe those of you who are interested in these topics see what we can do and how we can potentially collaborate or be useful to your studies. So as you well know, SARS-CoV-2 infection only sometimes causes COVID-19, and only sometimes severely, and only sometimes very critically. There's a huge variability. And that variability was already identified in the earliest cases in China, that COVID disease is really predominant in the elderly, and it drops off dramatically in, so this is in pink, hospitalizations, and in red, ICU admission, and black is death. And while some young people require hospitalization, it's very rare that they actually are admitted to the ICU or potentially die. There has been recent studies that have identified, particularly in New York, cases of young kids who develop a severe inflammatory disease. It's still a fairly rare phenomenon, but it's still notable that that is also something that exists. So what causes this variability? Well, genetic studies have identified two places in chromosomes that seem to have some significance, but it's probably not the key determinant of whether you develop COVID disease or not. Probably the key determinants appear to be conditions that prime for inflammation. And on this chart that describes young, middle-aged, or older people with a variety of different conditions, I think it's most remarkable to me is that the young who are obese or who have chronic lung disease are more likely to develop disease. I think hypertension and cardiovascular disease is predominant in the old, but I think there's a lot of those conditions are simply very prevalent in old people. So what I find striking about this graph is really the obesity and the chronic lung disease. And those are conditions that prime for inflammation, particularly in the lung. And so here is a recent study in Nature Medicine that identified two patient cohorts, and I'll tell you who they are later. There's a patient cohort that does not have significant IgG levels, and a patient cohort that does have significant IgG levels that develop adaptive immunity. And as you would expect, the ones that develop adaptive immunity knock down the virus faster, duration of viral shedding here, knock down virus faster than those that don't develop antibodies. And what's striking is that these cohorts were actually first defined as those that have symptomatic disease. So it's the ones that develop antibody were actually identified as symptomatic disease versus asymptomatic disease, indicating that the role of inflammation, of course, leads to antibody, but also symptoms. And those symptoms are driven by inflammatory cytokines like IL-6 and IL-2 and IL-15. So when virus infects cells, a number of things are produced, inflammatory cytokines and, of course, interferons. There's both antiviral and inflammatory responses, which are often described as basically the two arms of innate immune responses. And in this cartoon, it's described that those responses may be phased, that there's first an antiviral response that needs to be strong in order to knock down virus. And then with time, an inflammatory response takes over. And when that takes over, it can really lead to significant disease. And so this antiviral response is the one that also the virus is counteracting. So this is a cartoon out of Immunity that describes the signaling pathway by which virus is sensed by cells through receptors and then kinases and transcription factors. And each of these reactions is counteracted by coronavirus proteins that reduce the efficiency of an antiviral response. So in thinking about the variability of COVID-19, I think there's a couple questions that are of interest. And one is, how are innate immune response programs actually regulated and their specificity? To what extent can they be specific? And how do pre-existing conditions affect these responses? How is it that pre-existing conditions that seem to lead to disease severity? And that can be described with the concepts in the literature of trainability and memory of innate immune responses. And so we attempted to describe this because in some sense where there are innate immune sentinel cells are recognizing, of course, not just viruses, but all kinds of pathogens, 1,400 known species of human pathogens, and also tissue damage or damage associated molecular patterns that are the result of dying cells. And as macrophages, or generally immune sentinel cells, recognize these various substances, they produce not just antiviral and inflammatory programs. There's a lot more that they need to balance. Certainly there's cell intrinsic defenses, but even those cell intrinsic defenses must be pathogen appropriate, because in some cases, cell death needs to be prevented in order for the macrophage to function as an anti-bacterial fighter. Or cell intrinsic defenses may also actually be beneficial in triggering apoptosis to eliminate intracellular pathogens. So this is an example. Then, of course, the immune sentinel cells coordinates the local immune response, by which cytokines and chemokines are secreted to warn neighboring cells and bystander cells, and help with tissue remodeling, and potentially also undergo necroptosis to amplify inflammation in the local environment. And then coordination and initiation of a systemic immune activation, by which inflammatory cytokines are secreted that then lead to a systemic immunity and adaptive immunity. So in thinking about this, we sort of thought, well, maybe this is not just two arms of an innate immune response, antiviral and inflammatory. Maybe there is actually more. And of course, RNA profiling, first with microarrays by Rick Young's lab, identified some degree of pathogen-specific immune responses. Here, it's gram-positive bacteria that produce cluster A, or gram-negative bacteria that produce cluster D down here, but there's also a very common core response. And similar types of experiments were also done in my lab, where we could find different types of clusters. Interestingly, in these innate immune sentinel cells, like macrophages, or also tissue fibroblasts, but not B cells, whose specificity depends on their antibody, not their gene expression program. And more recent studies were done by also the Regev lab in dendritic cells, or in our lab in murine human macrophages. So in each case, you see patterns of expression or that are stimulus specific. And that was exciting, especially when you then recognize that each cluster, gene expression cluster identified by hierarchical or k-means clustering, correlates with gene ontology terms that indicate different functional outcomes. So for example, transcriptional control mechanisms in one cluster, or cytokine production in another cluster, or antigen presentation in a specific cluster. So these genes are produced or expressed in response to some stimuli, but not others. And so what we have done in recent years to try to describe how these genes are actually regulated. And there are 4 pathways that are almost sufficient to account for their regulation. So these are the 7 gene expression clusters identified. And four pathways may control the expression of these. For example, cluster A is primarily regulated by a MAP kinase type pathway, or AP1 pathway. B, C, and D primarily by NFKB, where D is also generated by the coordinated or synergistic function of NFKB and P38. And cluster E is controlled by NFKB and the IRF pathway. And F and G are really primarily IRF target genes. So we were able to map specific pathways of specific transcription factors to these gene expression clusters that are expressed stimulus specifically. And that type of model could be transferred to macrophage responses to a huge number of different ligands or live pathogens, and could account for their expression to some extent, but certainly not for all genes within each cluster. And so I'm highlighting the fact that here there's a lot of black lines indicating that the model does not fit the expression of these genes. And the percentage indicates how many do pass this kind of goodness of fit threshold. So many genes don't seem to be accounted for by the simple idea of four pathways. And the reason for that is that actually clustering itself, and I think Steve Smale has made that point for some time now, that clustering itself, the statistical correlations of this kind where genes that seem to have similar patterns of expression are put into the same cluster, that gives an underestimate of really the complexity of gene expression in macrophages. So for example, within this cluster B, where all the genes seem to behave similarly when you look coarsely, if you look in more detail, here are 3 genes from this cluster B. And you can clearly see that, for example, IL-10 is not expressed in response to RIG-I, or IL-10 is not expressed in response to VSV, whereas TNF and A20 or TNF-IP3 are expressed. So there is actually a much more heterogeneity in these clusters than within each cluster than this initial analysis suggests. So that prompted Catherine Xu recently in the lab to actually examine this in more detail by looking at not only at each gene, but also at each cell using single-cell RNA-seq. And single-cell RNA-seq maps here, each dot here is a cell that in this case derives from unstimulated conditions, or half-hour stimulated conditions, or 1 hour and 3 hours and 5 hours and 8 hours. And then on this t-SNE map, you can see the trajectory of cells as they are responding to LPS. And that trajectory is stimulus specific. If a lower dose of LPS is used, there is a slightly different trajectory for PAM3CSK or CPG, poly-IC, or TNF. And these trajectories are sufficiently distinct that when the data is used for a machine learning classifier, that you can distinguish them. And the 30 minutes, at the first time point, just after 30 minutes, most conditions can't be distinguished yet, except for poly-IC. So that actually cells responding to poly-IC within 30 minutes know or have expressed genes that indicate that they are responding to poly-IC. And within 3 hours, pretty much all conditions can be distinguished with a high degree of confidence, and at 8 hours even more so. So this study is really just nascent and just beginning, and more data needs to be collected. But I think it's going to be exciting to take a look at that. Now, how is it possible that these these immune sentinel cells can generate so many different gene expression programs that are highly stimulus specific. We only had 4 pathways that seem to play a role, and thinking about those 4 pathways is insufficient. And so one aspect that we did not consider previously is the dynamic control of these pathways. And so in work that recent graduate from the MBIDP, Ade Adelaya, examined macrophages responding in real time to different PAMs. And here is a movie of that, in which these macrophages in real time, and these are hours and minutes, are responding to TNF, and NFKB is labeled with a GFP variant. And you can see that it goes into the nucleus and comes back out and goes into the nucleus, leading to an oscillatory activity. Whereas sister cells, macrophages also, responding exposed to LPS, not even a very high dose of LPS, are responding, but NFKB remains in the nucleus and remains rather non-oscillatory. And so there substantially different dynamic control between these two. Now, data like this is complex and can be visualized through a heat map in which every row here is an individual cell whose NF-κB activity in the nucleus can be tracked over time, over 12 hours in this case. And you see in response to TNF, all of these cells seem to respond in an oscillatory manner, not synchronized, but each one is oscillatory. Whereas in response to LPS, most cells are non-oscillatory. The activity remains in the nucleus for some period of time. And so similar experiments were done for 5 different PAMP ligands at 7 different doses, and each time collecting many, many cells, many time points. We get a lot of measurements. And so we have data like this and heat maps for different stimuli at each of these different doses. And so, and all of this data, analyzing all of this data, led us to identify 6 dynamical features in NF-κB activity that seem to be deployed stimulus specifically. For example, the speed of activation is particularly high in the mid-88 ligands, TLR2 and TLR4, that are responding from the surface. The amplitude is higher with some ligands than with others. Some ligands lead to oscillatory activities and others to non-oscillatory activities. The total activity of the response can be higher or less. The total duration of the activity can be longer or less. And how much of the activity is front-loaded, is early versus distributed or late, is also different. So these are dynamical features that actually convey information to the nuclear target genes. And as a result of that, just using those dynamic features, the nuclear target genes know with a pretty high certainty, this is again a machine learning classifier, that TNF was the stimulus that is on the outside of the cell, or PAMP-CSK was the stimulus that was on the outside of the cell, or CPG or LPS or poly-IC. If they only have information on the dynamics of NF-κB, the nuclear target genes will know what happened on the outside with some degree of certainty. And of course, there's also some possibility for confusion at that point. If you're simply considering host cytokines like TNF versus bacterial PAMPs or viral PAMPs, the degree of certainty that nuclear target genes have is higher. Now, NF-κB is, of course, really important, but MAP kinase P38 is similarly important, and ongoing projects by postdoc in the lab, Stephanie Lücke, is investigating that. And we, again, find stimulus-specific dynamic control of P38. And these studies are ongoing, and I think we're going to find something interesting also how that is coordinated with NFKB. Similarly, Hatera Wilder, another talented postdoc in the lab, is looking at the interferon response factor, IHG3. And even this factor, responding either to the type 1 interferon or type 3 interferon, shows stimulus-specific dynamic control. Both are going up to the same level initially, but then what happens after that is different for type 3 versus type 1. And whereas both stimuli activate gene expression of some genes, there's some other genes that are highly type 1 interferon specific and are not induced by type 3. So, so these dynamics are correlated with gene expression, but by using mathematical models, we can also show that they actually explain gene expression by thinking about the kinetics of chromatin opening, of promoter activation and recruitment of the pre-initiation complex to the synthesis of RNA and the decay of the RNA. And by developing models, we can therefore improve how many genes can be fit to, can be accounted for by not only the 4 pathways, but when we consider the dynamics of signaling in addition. And here this study was published recently in Cell Systems about NFKB control of gene expression. So as a summary here, my first summary is simply that immune sentinel cell responses are really highly pathogen specific, much more complex than just the category of inflammatory and antiviral. And that requires, you know, a much more sophisticated analysis to really make progress and give insight. And the second summary is that those responses are generated by the combinatorial action of 4 pathways, and that the stimulus-specific dynamics of each pathway, and that is described sort of in the systems biology literature as either using combinatorial coding, where different genes might be subject to the control of multiple transcription factors that are either in an OR gate or AND gate or configuration. But each of these pathways also is dynamically controlled. And so there's also temporal coding, where different dynamics of transcription factor activity can be distinguished by genes depending on how their chromatin dynamics are regulated. So I think we're the field of innate immune responses, and I think, and my lab is having a lot of fun clarifying to what extent innate immune responses can be specific and how that specificity is regulated. But how are pre-existing conditions then affecting these responses? And in the field, that is described sometimes as simply context-dependent responses, microenvironment dependent responses, or also to what extent the context can train the responses, and the trainability of innate immune responses, and to what extent that trainability can be memorized, can provide immunological memory. And all of these concepts are reasonably well established, but how they really work remains active research, and also what their functional consequence is. So here's work that Quen Cheng, when he was still doing his PhD in his lab, he's a staff fellow, did, and where he profiled gene expression responses in human macrophages together with the modeling lab, and found stimulus-specific responses, you can see. But when those same macrophages were primed or pre-exposed with interferon beta, the responses were slightly different. And with interferon, and when they were pre-exposed with interferon gamma, those same stimulus responses were different again. And on the left side, the map indicates what the differences were when cells were interferon beta primed or interferon gamma primed. Sorry, I missed this one here. So you see that what I want to emphasize here is that both beta priming and gamma priming can lead to an increase or a decrease of gene expression. And that those differences are gene specific and stimulus specific. So these increases seem to apply to almost all stimuli, but this decrease applies to only one stimulus and not the other stimuli, and only to some genes and not all genes that are induced by poly-IC. And similarly for interferon gamma priming. So there's an incredible amount of sophistication and complexity in being in these innate immune cells, or human macrophages in this case, in being able to adapt, adjust their gene expression responses to different ligands and to different contexts or histories of exposure, in this case, beta versus gamma. And Quen mapped actually some of these changes, but some of these changes actually affect regulators. So gamma priming leads to a decrease in TLR2 expression, which of course affects then the response to PAM3CSK. Or beta increases the expression of TLR3, which affects the response to poly-IC. And gamma will reduce the expression of TLR3. And there are many, many other changes that then lead to a change in transcription factor activation after cells have been pre-exposed. So this is just beginning to illustrate the complexity. We certainly don't understand precisely how this network works, which is why we have this rather ambitious plan to develop a virtual reality or a computational model for the macrophage. But in addition to changing the network, the signaling network, priming by beta and gamma also changes the epigenome that then interprets those transcription factor activities. And Quen identified thousands of chromatin regions that show changes in chromatin accessibility in response to beta and gamma. And here you see some examples. So CXCL10 is not affected a whole lot by beta, but is affected by gamma. And whereas this gene GAM shows reduced chromatin accessibility in response to beta and very little effect by gamma. And those changes in the epigenome then are correlated with differences in responses, such that gamma priming here, which increases chromatin accessibility, leads to an increased and faster response, whereas here, reduction in the chromatin accessibility leads to a reduction in gene expression responses subsequently. So similarly, Supriya Sen, project scientist in the lab, is profiling how NFKB responses themselves, in this case, in response to TLR8. TLR8, of course, is a viral sensor, activates NFKB in naive macrophages in this manner. Beta pre-stimulation of prior exposure with interferon beta has very little effect, but prior exposure with gamma has a huge effect. And IL-10 has some effect, but IL-4 and IL-13 particularly convert the initial non-oscillatory response into an oscillatory response. And so there are subtle and interesting changes that occur by priming with cytokines. And going back to some of Ade Adelaya's work, he examined NFKB responses not by priming with cytokines, but in an autoimmune disease model in which macrophages are in an environment of autoimmune, auto-inflammatory and autoimmune microenvironment. And that leads to NFKB dynamics that are altered. Here I'm simply showing the dynamic features that I just talked about earlier, the peak amplitude, the speed, oscillatory, non-oscillatory, and how they are different in healthy macrophages between TNF and LPS. But in macrophages derived from these Sjogren's animals, some of these features are the same. They still are faithful, but others are messed up and therefore lead to potentially to stimulus confusion. This one and this one are all the same and are not distinguished anymore in response to these three stimuli. And so that, whereas in healthy macrophages are very good at distinguishing TNF, LPS, and poly-IC based on NFKB dynamics, in these Sjogren's model-derived macrophages, there's a lot of confusion between TNF and LPS, meaning that if cells are actually seeing TNF, they communicate to the nuclear target genes as if they were seeing LPS, which might be then a reason for this continued autoinflammatory phenotype. So this inflammatory history also alters the de novo enhancers, as can be, also generates de novo enhancers. So Quen examined the epigenome looking at an enhancer mark, H3K4 monomethylation. And that enhancer mark, it shows increases at a few hundred chromatin regions, especially in response to LPS and poly-IC for these. And then for these other couple of hundred other chromatin regions, not so much in response to poly-IC, but in response to LPS, CpG, and PAMP-CSK. It turns out that these two clusters of chromatin regions, some of the top ones are enriched for ISRE elements, and indeed knocking out the interferon pathway also knocks out the ability to generate de novo enhancers. But in the bottom cluster, they show enrichment for kappa B sites, and those are unaffected by knocking out the interferon pathway. So these are NFKB driven. But what was curious is that even though TNF activates NF-kappa-B, it does not generate de novo enhancers. De novo enhancers are only generated by PAMP-CSK, CpG, and LPS. So what do those have in common? Well, PAMP-CSK, CpG, and LPS lead to non-oscillatory NF-kappa-B, whereas TNF and poly-IC show more oscillatory NF-kappa-B. And the oscillatory NF-kappa-B does not activate de novo enhancers, whereas NF-kappa-B is capable of creating de novo enhancers when it's non-oscillatory. How might NF-kappa-B be able to do that? It does so by opening chromatin as detected by ATAC-seq when NF-kappa-B is non-oscillatory. Here is actually using TNF, which leads to oscillations, or in the mutant, TNF leads to non-oscillations. So, whereas in the control, these regions are not opened very much. When NF-kappa-B is non-oscillatory, those regions get opened. And when they're opened, they can acquire enhancer marks. And while the opening is transient, when the stimulus is washed away, chromatin closes again, the enhancer mark remains. And so thereby these enhancers, even though the stimulus is transient, the enhancers remain, and those enhancers by remaining there, impart some level of innate memory. And that innate memory results in some of those regions now responding to a stimulus like TNF that they previously did not respond to. So this is all in vitro, but of course, much more important work has been done in vivo, for example, by our colleague Michael Yeaman, who described that innate immune memory is also really important in contributing to the host defense in Staph aureus. And that was a very nice paper published a couple of years ago. And there are several other examples probably just like that. This innate immune memory, one version of it, of course, is tolerance that is broadly described, where one-time sublethal injection with LPS leads to macrophages that, even when taken out of the body, now produce less cytokine than they used to. And what this paper showed a few years ago from the Sternberg lab is that actually this innate immune memory can also be reversed, in this case by priming these cells first with beta-glucan. And then they lose their memory, if you will. They revert back to a state where they respond to LPS more strongly. So this idea of innate immune memory is one that is interesting, and how long it lasts is certainly an important topic, and how it can be manipulated in a useful way is a really important interest. So my summary here then is that healthy innate immune responses involve many functionally distinct gene expression programs, not just inflammation and antiviral programs. They are highly specific to stimulus and also to tissue context. And those responses and the specificity of those responses are affected by prior exposure history, and therefore cytokines associated with pre-existing conditions. And so in that regard, the hypothesis would be a great grant proposal at some point, is that the state of innate immune cells may in fact predict COVID severity. So given that I don't have any data to support that hypothesis other than the background, I will leave it at that. I want to thank particularly my lab. You know, we always say, so many emails I've gotten and written say at the end, stay safe and sane. Well, it's the lab that keeps me sane during this time. And so thank you not only for this great work, but also for keeping me sane. With that, and thank you to you, the audience.
Great, thank you so much, Alex. That was a really, really, really beautiful talk. So I guess now we'll open the floor to questions. You can either type your questions in the chat box and I'll read it out to Alex, or you can unmute yourself and ask him directly.
Hello, Alex. This is Srivatsan from the VA.
Yes.
How does the mutations affect this innate immunity? If you think it is a prior cytokine or innate immunity is there, now the virus mutates itself. You know, in influenza it does it. What happens in COVID? Certainly, the death rate in the US and Brazil is very high compared to some of the Asian countries. Can that innate immunity explain, or is a mutation that's doing the innate immunity?
So you're referring to mutations of the virus.
Yes.
I'm sure. I mean, obviously, flu mutations are primarily to avoid adaptive immune functions. In my mind, that's what I understand, and that's how strains of flu are characterized. And I imagine similar similar mutations will be identified in SARS-CoV-2. It just hasn't been long enough yet. In terms of manipulating the innate immune system, that has to do probably more with pathogenicity rather than, I don't know of any studies yet of coronaviruses differing in the way they interfere with the innate immune system. But I'm sure that's going to be also a factor. Yes, it's a good question.
Yeah, the reason is there is a lot more death around the United States and Brazil compared to, and of course, Italy and Spain, compared to the Asian countries. There has to be some difference in immune response or cytokine response. I don't know what it is, inflammation.
Mm-hmm.
You are differentiating inflammation from the innate immune response. Did they have prior some COVID-like infections in the past that now they are getting a better innate immune response in the Asian countries?
Yeah, I think if you're comparing different countries, there's so many different variables. I mean, starting just with the reporting differences, and the data of, so I think it's hard to conclude with reliability, given that we're not testing, most countries are not testing nearly enough people to get reliable numbers. But the other aspect, of course, is that the innate immune system is very much affected by prior exposure, hygiene hypothesis, food, all of these other different things are going to make a difference. And I saw an interesting comment in the chat about the BCG vaccine might possibly playing a role. But that is, I think it's going to be a really active area to look into. I agree.
Thank you.
So Alex, let's go to the questions in the chat. And I see the BCG questions are kind of related, right? So I'll group them together. What is the effect of different strains of BCG to COVID-19? And then followed by, in most Asian countries, BCG vaccines mandatory and death rate in these countries is low. Do you think BCG vaccination is helping?
Ooh, I wish I knew the answer to that. I really don't know. I don't know how to begin to address that question. I'm sorry. I have a feeling the person who's asking the question might know more about that than I do. Does anybody want to, could be a discussion. If anybody has an idea, maybe somebody else can jump in.
I guess no further comments on that. I mean, I think it's a very interesting question. And this is related to the data you were showing towards the end, right? With the BCG priming.
Right. That was beta-glucan. That wasn't BCG. I think wouldn't that primarily be an adaptive immune effect with BCG? I'm not sure. Yeah, I'm unclear what the connection could be.
One comment that just popped out is life attenuated BCG appears to respond differently to COVID.
Mm-hmm.
Okay, another question from Liying Zhang. Alex, could you comment on the effects of androgen and anti-androgens on the innate and acquired immune system?
Well, we haven't studied that, and I haven't kept up with the literature specifically, but certainly in general that there's a rewiring or a alteration in the expression levels of many of these signaling molecules. There's also androgen receptor can synergize with some of the NFKB members in activating expression. but I don't have a clear idea how that might be relevant or be important for COVID and and the sex differences that we see in disease. I think generally the innate immune system often has stronger responses in females than in males. Antiviral responses tend to be stronger in females than in males. And so I'm not surprised that we see sex differences. But I don't think it's been looked at very, very clearly, specifically for COVID.
Hi, Alex. This is Lily Zhang. Hi, Melanie. Thank you for reading my question. So, are there any gender differences for the NF-κB pathway?
Yeah, great question. I don't think so. There's no sex differences, no gender differences for NF-κB. The differences that have been documented are upstream in the receptors for some of the PAMPs, the receptor expression of some of those receptors for viral recognition is elevated in some of the, and they're X-linked. And so, but for the NF-κB pathway itself, there are no differences. None of those proteins are X-linked.
Thank you.
So Alex, can I jump on, jump on that question? I was curious with the oscillatory phenotype, do you think there could be an age dependency?
Yeah, I think so. So I think age and inflammation go hand in hand, right? Sometimes more severely than less severely. And so that does play a big role. Absolutely. And we haven't compared macrophages from old and young animals or people yet, but we have looked at hematopoiesis and the control of NF-κB activity in hematopoietic progenitors, and we see substantial differences there that help explain why you see hematopoietic decline in age. And that's a completely different study from what I've shown here. But I think inflammation associated inflammation as associated with aging clearly has a big impact. It has a big impact on the epigenome, and it has a big impact on the signaling responses, and therefore gene expression. Yeah.
That's cool. Yeah, I was curious because probably the macrophages that you guys isolate from the mice usually are from a specific age group, or that you wouldn't maybe look at very old or very young or very different in age. Okay, I think do we have another question? Is it possible that the children get less severe form of COVID because they receive series of vaccinations early on?
Well, I doubt that those vaccinations provide protection against COVID. I think if it is surprising, kids are capable of very strong inflammatory responses, but they don't seem to, there's less detriment to, high fevers. So there are protective mechanisms that kids have to inflammation. So but why kids are protected from COVID largely, there are these outlier children, and it's not a small number, who develop very, very severe inflammatory disease, Kawasaki disease. And I think people are studying that, but I don't know exactly what's going on there.
Is it the thymus that comes into play in the children? Is that the reason? Because that might be getting immune response compared to the adults.
Maybe.
Any other questions?
Well, if anybody wants to study innate immune responses with us, we're welcome any interest. And I try to describe some of the tools that we have, and we can apply them to disease models that you may have. And so look forward to that.
Oh, Alex, one last question from Tracy. The autoimmunity data you present is very interesting. Do you know if there are any studies on the relationship between autoimmune diseases and COVID severity?
That's a really good question, and you would expect that there are relationships, but I have not seen any correlations there. Maybe I haven't looked specifically, but whenever I see charts of what the pre-existing conditions are, I don't see autoimmunity as being one of them.
But I think it would definitely be very interesting to do like the study you show, right? In cardiovascular disease or diabetes kind of context. Is the response also confused?
Yeah.
Great. Thank you so much, Alex. That was really interesting and stimulating discussion.
Great. Thanks very much.
Yeah. And I guess maybe before we close, I just want to quickly invite anyone to suggest seminar topics that you think will be interesting and relevant, or any potential seminar speakers you want to nominate to talk. Please let us know, me or Bridget, or just type it in the chat room right now. So, great. Well, thank you. We'll see you next week. It's going to be Vithi that will talk to us about antiviral drug discovery. Have a good week.
Thanks very much.
Thank you.
Cool. Bye.